Welcome to Mythosium, the channel where we take the most intricate scientific ideas and make them feel beautifully intuitive. Today we're venturing into a hidden world, one that exists inside every cell of your body. It's a world filled with tiny molecular machines operating with extraordinary precision, working relentlessly to keep you alive. These structures are so small that billions of them could fit within the period at the end of a sentence, yet they perform tasks as complex as the most advanced machines ever designed. Right now, as you listen to this, a vast number of your cells are undergoing a beautifully choreographed process known as cell division. This process allows your body to replace cells that have aged, been damaged, or reached the end of their programmed lifespan. In an adult human, tens of billions of cells die each day, and just as many are created. This astonishing equilibrium is maintained to a sophisticated system that copies, packages, and distributes genetic information with astonishing accuracy. At the heart of this process lies DNA, the famous double helix that encodes the instructions for life. Each strand of DNA is composed of a sugar-phosphate backbone paired with 4 chemical bases: adenine, thymine, guanine, and cytosine. The 2 strands run in opposite directions, a detail that might seem trivial but becomes essential when cells prepare to divide. Before a cell can split in two, it must create a complete copy of its entire genome, ensuring each daughter cell receives identical genetic material. This replication process begins when specialized molecular machines latch onto specific points along the DNA. The first of these machines is helicase, a remarkable enzyme that unzips the 2 DNA strands by spinning rapidly and breaking the hydrogen bonds between the bases. Helicase can unwind DNA at speeds comparable to jet engine rotations, separating the strands so that each can act as a template for construction of a new complementary strand. However, replication occurs in one direction along the DNA, and because the strands run opposite ways, the 2 templates must be copied using different strategies. One strand is synthesized continuously, following helicase as it opens the double helix. The other strand, oriented in the opposite direction, must be assembled in short fragments that are later stitched together. This process involves an array of enzymes, each performing a specific function, from adding new nucleotides to sealing the fragments into a continuous chain. By the end of this phase, the cell has 2 identical DNA molecules, each only a few nanometers thick but several centimeters long. To prevent this long molecule from becoming tangled, it wraps itself around spherical proteins called histones, forming bead-like structures known as nucleosomes. These nucleosomes coil into thicker fibers, which then loop and fold into complex shapes. When fully packed and prepared for cell division, these structures become visible under a microscope as chromosomes. Each chromosome contains 2 identical copies of the DNA, known as chromatids. These chromatids remain joined until the moment of separation during mitosis, but separating them is far from simple. It requires an elaborate apparatus composed of additional molecular machines, each performing highly coordinated mechanical and chemical tasks. One of the most sophisticated structures involved is the kinetochore, a dense cluster of hundreds of proteins assembled on each chromatid. The kinetochore serves as the anchor point where microtubules, long, rigid structures that extend across the dividing cell, attach and exert forces on the chromatids. These microtubules constantly grow and shrink, a dynamic behavior that helps align the chromosomes along the central plane of the cell. As the chromosomes are positioned, the kinetochore performs a dual function. It physically stabilizes the attachment to microtubules and also acts as a checkpoint. If a chromatid is not properly attached, the kinetochore emits a biochemical stop signal that halts the division process. This ensures the cell does not proceed until everything is precisely aligned. Once proper tension is detected across the chromatids, the kinetochore prepares for separation. But switching off the stop signal is not as simple as flipping a switch. Instead, the signal is physically removed. Molecular motors known as dyneins attach to the signaling proteins and carry them away along the microtubules. Dynein's distinctive walking motion, powered by the energy stored in ATP molecules, transports these signals toward the poles of the cell. This makes way for separation to begin. At the same time, kinesins, another class of molecular motors, walk in the opposite direction along the microtubules. Their movement is equally graceful and efficient, allowing them to carry cargo, rearrange microtubule structure, and help guide chromatids. The coordination between kinesins and denins resembles a microscopic roadway system, where traffic flows in both directions along dynamic molecular tracks. Once the checkpoint is cleared, the chromatids are pulled toward opposite ends of the cell. The exact mechanism behind this movement remains one of the most intriguing unsolved questions in cell biology. While microtubule shortening contributes significantly, subtle interactions between motor proteins, structural changes in the kinetochore, and tension-driven dynamics all play essential roles. As the chromatids reach their destinations, the cell prepares to split. New membranes form around the two sets of chromosomes, re-establishing nuclei. The cytoplasm divides, organelles are distributed, and the two daughter cells begin their independent lives. This entire choreography, from DNA replication to final separation, takes place in about an hour in mammalian cells. Some cells reproduce much faster, others slower, but the machinery is universal. The remarkable part is not just the timing, but the precision. A single mistake during division could lead to cell death or genetic instability. The fact that trillions of cell divisions occur in a human lifetime with such accuracy highlights the extraordinary reliability of these molecular systems. Each part of this process is carried out by machines that assemble themselves automatically, using nothing more than the chemical properties encoded in DNA sequences. They operate at speeds, scales, and levels of precision far beyond anything currently achievable with human-made nanotechnology. These machines are not only elegant, but efficient. They demonstrate how complex behavior can emerge from simple interactions when coordinated across many components. They are powered by fundamental physical forces, the attraction and repulsion of charged molecules, the spontaneous formation of hydrogen bonds, the release of energy through ATP hydrolysis, and the self-organizing tendencies of proteins. Perhaps the most astonishing aspect of this molecular world is that it raises questions about what technology could become. If biology can produce machines that walk, clamp, rotate, pull, and assemble, all within the confines of a single cell, what might be possible if humans learn to harness similar mechanisms? The idea of nanobots capable of repairing tissues, clearing blockages, or delivering precise treatments no longer feels purely fictional. Nature has already demonstrated the blueprint. It's only a matter of learning how to replicate or adapt these principles. Even today, scientists continue discovering more about how these systems operate. Some molecular machines remain only partially understood. For example, the exact process by which chromatids are pulled apart involves forces and structural changes that researchers are still unraveling. The kinetochore, despite its central role in cell division, contains layers of complexity that have only recently become accessible using advanced imaging techniques. Every discovery in this field highlights how much remains unknown. The microscopic landscape inside the body, though studied for more than a century, still holds countless mysteries. The deeper researchers look, the more intricate patterns they find. Feedback loops, structural motifs, mechanical linkages, dynamic rearrangements, and networks of interactions that resemble the internal workings of a factory. Yet, these machines are not factories in the industrial sense. They are flexible, adaptable, and governed by principles that blend chemistry, physics, and information theory. They reorganize themselves when damaged, they adjust to local conditions, they respond to signals with exquisite timing. Understanding these systems is not merely a scientific exercise. It is a way of appreciating the hidden complexity that makes life possible. The smooth functioning of tissues, the healing of wounds, the growth and renewal of the body, all of it is powered by machines so small and so precise that they operate beyond the realm of sight. Here at Mythosium, we explore this world not just to marvel at its elegance, but to reveal how deeply interconnected all living systems are. The molecular machines inside every cell tell a story of evolution, resilience, and innovation, a story that continues to unfold with each new discovery. If you enjoy learning about the hidden mechanisms that sustain life at its most fundamental level, be sure to subscribe. There are countless microscopic wonders waiting to be explored, and together, we'll continue uncovering the extraordinary machinery inside us all. Thank you for watching, and stay curious.